Hello everyone, my name is Christina Welch and I'm a master's student at the University of Georgia in the Animal and Dairy Science Department. And today we're going to talk about Clostridium aminophilum. So what is Clostridium? Clostridium is a diverse group of bacteria from the family Firmicutes, which include both harmful and beneficial bacteria. Some of the pathogens present in this group include Clostridium botulinum, which is the causative agent for botulism, and another one is Clostridium difficile, which causes many gastrointestinal problems in humans. And one that's found in cattle is Clostridium shovii, which is the bacteria that causes blackleg. Although these bacteria are all in the same group, they generally are quite different from each other. However, for the most part, they're anaerobic, meaning they like to be in environments with no oxygen, and they also form endospores, which, as you can see from the picture on the right, is the circular part on the rods. And this is what causes Clostridium to be really hard to kill. So the bacteria I'll be focusing on today is Clostridium aminophilum. It was discovered in 1993 by Bruce J. Pasteur, and he described it as an irregularly shaped gram-positive rod that kind of looked like a football, so therefore it got the nickname the F-bug. It was 1 micrometer wide and 1.5 micrometers long. And he also said it was a non-motile bacteria that was an obligate anaerobe, which gave it a competitive advantage in the rumen, as it's able to use oxygen while also living in an environment without oxygen. One unique thing about it is it's a saccharolytic bacteria, meaning that it's unable to utilize carbohydrates, and however, it grows on amino acids and peptides instead. Now, Clostridium aminophilum belonged to a group of bacteria found in the rumen called obligate amino acid bacteria, or another name for them is hyperammonia producing bacteria. The other two bacteria found in this group include Clostridium stricklandii and Streptococcus anaerobus. One unique thing about them is they have an extremely high affinity for amino acids. And one of the reasons why it took scientists so long to identify them in the rumen is they are found at extremely low numbers. However, they're able to deaminate amino acids 20 times faster than other ruminal bacteria. So although they're found in really low numbers, they make a very large impact. Since obligate amino acid bacteria are able to degrade peptides so quickly, it's no surprise they're involved in the overproduction of ammonia in the rumen. The breakdown of amino acids in the rumen is a very wasteful process that leads to an excess of ammonia in the animal, which is eventually lost as urinary urea. If the energy is limiting in the animal, or if peptides are broken down faster than amino acids are taken up, then the nitrogen that is found in the rumen turns into excess ammonia, which then causes the nitrogen retention to become an inefficient process. Clostridium aminophilum is responsible for 9% of the protein lost through feed, and this causes the producer to have to pay more for such an expensive ingredient. Since obligate amino acid bacteria are responsible for so much protein lost in the rumen, it's important to find a solution to combat this problem, and Meninsin provides an alternative solution to using antibiotics. Meninsin is an ionophore that targets gram-positive bacteria. A study that was performed at Cornell University by Krause and Russell looked at the effects that Meninsin has in the rumen. It showed that it decreases ammonia concentration while still maintaining VFA production and decreasing the acetate to propionate ratio, which means that there's more energy available to the animal. The study was divided into two parts, and in the in vitro section of the study, they looked at growing the 3 obligate amino acid bacteria in co-culture with Meninsin. They found that Clostridium stricklandii and Streptococcus anaerobus could not grow in batch culture containing more than 0.5 macromolar of Meninsin. However, Clostridium aminophilum was able to grow in as high concentration as 1.5 macromolar of Meninsin. When they moved it to an in vivo study and they looked at the effect it has in the animal, they found that Clostridium stricklandii and Streptococcus anaerobus could no longer grow. However, Clostridium aminophilum not only continued to grow, it also increased its population when monensin concentrations were as high as 7.2 micromolar in the rumen. This information combined with another study at Cornell, which found that Clostridium aminophilum could retain its resistance to monensin for at least 28 generations, even after monensin was no longer present, means that there needs to be an alternative solution to monensin to treat Clostridium aminophilum. The first alternative solution to using monensin in order to inhibit the growth of Clostridium aminophilum populations in the rumen is by using a bactericin. The rumen is a very competitive environment, and in order to gain a competitive edge, bacteria produce things called a bactericin, which target and kill off other bacteria within the same niche. Bovicin HC5 is a bactericin produced by Streptococcus bovis strain HC5. A study produced by Mantovani and Russell at Cornell University found that when included at 12.5 activity units per milliliter, not only was ammonia concentration decreased, but it also strongly inhibited the growth of Clostridium aminophilum. And at sublethal levels, which were 10 activity units per milliliter, the growth of Clostridium aminophilum was slowed to levels that were indicative that this bacteria would not gain a resistance to this bactericin. The second alternative solution to using monensin is the use of hops. Secondary plant metabolites have been used to control bacterial fermentation for a long time. An example of this is using hops in beer as a preservative. A study performed at the USDA Agriculture Research Service in Lexington, Kentucky found that lupulin found in hops was acting on the cell membrane of hyperammonia producing bacteria and showed a decrease in the ammonia production. The study also found that in the presence of hops extract, hyperammonia producing bacteria were depleted of their potassium used in their transmembrane potassium gradient. Therefore, they were unable to grow and their populations decreased. Now, the last one is not necessarily an alternative solution, but it's a very important topic in animal agriculture today. One concern in society is antibiotic resistance. So, a study conducted by Cornell University looked at Clostridium aminophilum that was resistant to monensin to see if this resistance carried over into other antibiotics. One reason for this is many countries have banned the use of antibiotics and also ionophores in animal agriculture. However, the study found when monensin-resistant strains were tested on their resistance to 15 other classes of antibiotics, Clostridium aminophilum was still susceptible to all but one class, which was bacitracin. So although they can retain their resistance for 28 generations to ionophores, this doesn't necessarily mean that they're cross-resistant to antibiotics. This concludes our discussion of obligate amino acid bacteria, specifically Clostridium aminophilum. These are the references that were used in this presentation, so if you have any other questions, you can follow the links here. Thank you so much for watching.